So thank you everybody for joining us today. So today is the first meeting that we had for the Meet the Scientist webinar series. And as a reminder, so the goal of this webinar series is to actually get an opportunity for junior investigators to learn more about the exciting research that is happening in Alzheimer's disease and also learn more about potential career opportunities that are available out there. So let me see if I can move. So today I have the pleasure and honor to be hosting the HABS, that is the Harvard Aging Brain Study. But before we go to all the invited panelists, I just want to remind everybody about the two next Meet the Scientist series events that we have. So in November 10th, we had Meet the Vanderbilt Memory and Alzheimer's Center at 4 p.m. And in December 3rd, at 4 p.m. as well, we had Meet the Healthy Brain Project team. For information and registration, you can go to the website, and registration is now open. So please save the day and try to attend as many of these meetings as you can. I think there is a great opportunity. So today, as I mentioned, we have a Meet HABS, that is a Harvard Aging Brain Study. This is a very well-known group for many of you, but I think that today is an opportunity for you to learn more about some of the investigators and also ask questions that you may have. So, so there is a QA option, so feel free to send questions as they are presenting. I will do my best to go to some of the questions at the end. So meeting that, so we meeting the Harvard Aging Brain Study today. So I'm gonna go through some of the members of this team. It's a very big and rich team. And we have the leadership, Dr. Reisa Sperling, Dr. Keith Johnson, and Dr. Doreen Rentz. So I'm not gonna name all the faculty, but these are the names of the HABS faculty. Some of them are gonna be with us today. I'm gonna be presenting some of their work. And we also have postdocs in the Harvard Aging Brain Study, and the same, these are their names. Unfortunately, because of time, we cannot have everybody presenting, but it's an opportunity also for you to get to know who they are. And the same for the HABS coordinators and support staff. So this is also a very large list, but the same, like also an opportunity to recognize some of the names as well. So now I'm gonna pass it to Dr. Reisa Sperling, that is one of the co-principal investigators for the Harvard Aging Brain Study. Dr. Sperling.
All right, well, thank you so very much. And I wanna first thank you, Jacqueline, for setting this up. What a great thing, and as an honor to be the first group who's meeting people virtually online. So let me go to the first slide real quick. So I just want to set the stage a little bit for the Harvard Aging Brain Study and say that although today we're really going to be focused mostly on the research that's going on within HABS, the Harvard Aging Brain Study, I want to put it briefly in the context of what we're really all about trying to do. And that's really working together to understand healthy brain aging and promote that, but ultimately to prevent Alzheimer's disease dementia. And so there aren't many of us who work in HABS also work at the Center for Alzheimer Research and Treatment. And there, as part of CART, we call it, we also run a lot of prevention trials. And we are also looking for people who are interested for clinical fellowships and people who want to work in clinical trials, as well as today we're really going to be talking about the science that provides the rationale for these prevention trials. So next slide. So the Harvard Aging Brain Study was started by Dr. Keith Johnson, who, full disclosure, I am married to, even though we're sitting in two different rooms here. We really wanted to try to study people who start out as normal and understand what keeps them resilient and, unfortunately, what puts them at risk for developing Alzheimer's disease, especially focused initially on amyloid and then later on tau, and now many other factors that contribute to that. And we are one of the best things about HABS, I think, is our participants. We have this very dedicated group of participants, many of whom have now been in the study for 10 years. And we just got renewed in an NIH program project to be able to follow them for 5 additional years. So this is just to demonstrate the data we had. This is actually already a bit out of date because we filled in a few circles in 2020, even despite the pandemic. But these are individual participants showing them as they've moved through the project. And again, we have more than 2,500 cognitive test administrations and over 1,700 MRI. And I think we're now up to 900 amyloid scans over time, as well as FDG and tau PET. This isn't to mention blood tests and other kinds of cool things we're doing that you'll hear about from the amazing team here. I also want to say that I think one of the best things is working together in environment, and you'll hear from these folks over time, but it's an honor to be able to work with amazing junior and not so junior anymore as they become faculty investigators. And we're really excited to have new people from all different backgrounds, multidisciplinary individuals join us, because the next 5 years are going to be even better than the last 10. So I'll stop there and let others go on.
Thank you, Dr. Sperling. Now I want to invite Dr. Johnson to introduce himself.
Hi, I'm Keith Johnson. I will just very briefly echo what Reisa said, which is that we're excited to be involved with studying such an incredibly important and difficult problem, and to be able to do so with such a tremendous group of co-investigators, all of which would, of course, not be possible without our participants who really make this all happen. So I'm pleased that you're all listening, and I hope we can help you, help you learn something about what we're up to.
Thank you, Dr. Johnson. So now I'm going to pass it to Dr. Rebecca Amariglio.
All right, thanks, Yakeel. Yep, you can move to the next slide. So I'm Rebecca Amariglio, and both based at Brigham and Women's and MGH, as many of us are. I've been fortunate enough to have been working on the Harvard Aging Brain Study since its inception. So out of fellowship, happened to join right as I was ending fellowship, and now currently co-lead on the clinical core of HABS. My focus throughout has been on subjective cognitive decline, otherwise known as memory changes that individuals report despite performing normally cognitively on our neuropsychological testing. And so we probed this in the Harvard Aging Brain Study using a number of different questionnaires that we've been giving to participants over the many years to see if there are changes and what kind of memory changes they themselves are observing. Some of the initial work that we've done in HABS was looking at endorsement on these questionnaires with biomarkers that we've collected. So looking at amyloid and tau. So one of the first papers was seeing that individuals with elevated amyloid were also reporting higher amount of cognitive complaints on these questionnaires. And again, these are people who enter our clinically normal. So that was a finding that I think was valuable, that people themselves can notice these changes. We found something very similar with tau as well, with the Rachel Buckley leading on that effort. And then as the study has continued, we've looked at SCD, or subjective cognitive decline, as a risk factor for objective cognitive decline on neuropsychological testing. So alongside elevated amyloid, showing steeper decline. And I think now entering really interesting territory as we have 10 years of data to see how subjective cognitive decline evolves over many years. And so this figure to the farthest right, in people who are amyloid positive, seeing some initial increases in cognitive complaints on questionnaires, but then in around 4 years or so, seeing a plateau or even a decline, as maybe perhaps some individuals are moving towards having less awareness of cognitive concerns. So, and then we've also been sending these questionnaires remotely. So that's of a great interest of mine, as well as how can we continue to collect this information remotely, especially during these uncertain times when we're not necessarily seeing participants in person. And then the next slide. I did want to let people on the call know about an opportunity for budding neuropsychologists. So for those of you who might be trainees in clinical neuropsychology, you may have heard of the Harvard Partners Consortium in clinical neuropsychology, which is primarily focused on clinical training, with a small research component. And we've recently added a new track that we're launching this year more formally, where individuals would have the opportunity to both get board certified with clinical training at Brigham and Women's Hospital, but then also be able to work on research in the Harvard Aging Brain Study. So we really encourage people from all over to apply, and we hope to entice people who have an interest in Alzheimer's research and aging research, who are also looking to get clinical training at the same time. So that's.
Thank you, Dr. Amariglio. So I'm going to pass it now to Dr. Rachel Buckley.
Hi, yeah, if you wouldn't mind moving to my slide. Thank you, Yakeel. It's lovely to meet everybody on this call. My name is Rachel Buckley. I am an instructor at MGH in the Department of Neurology. I am the co-lead of the Data and Informatics team at the Harvard Aging Brain Study. And my focus of research is really on sex differences in risk for Alzheimer's disease, particularly in terms of resilience or vulnerability to amyloid and tau pathology. My main modalities that I work with are PET and MRI, markers of amyloid, tau, and neurodegeneration, but I've also started to move towards looking at cerebrospinal fluid and plasma fluid markers as well, with the main intention of trying to understand the earliest time in which sex differences really start to appear. And I'm now starting to really focus on why this might be. The direction that I'm starting to head towards is looking at whether menopause markers or sex hormones may show some association with this resilience or vulnerability. But I wanted to take a couple of seconds of my time just to also reach out to the group, people who are listening in on this call. I am looking for postdocs to join me and my team, my lab, in summer or fall of next year. Please feel free to reach out to me at my email, or you'll also find me on Twitter. I do like to retweet a lot of science. So just a disclaimer on that, but thank you so much for joining the call and listening in on our team. And any other questions beyond sex differences, feel free to reach out to me as well. Thank you.
Thank you, Dr. Buckley. So now I'm going to pass it to Dr. Jess Chadwick.
Great. Thanks very much, Yakeel, for organizing this, and to all of you out there who are joining us for it. It's a great project, so it's a lot of fun to talk about. Can you go to the next slide, please? So I'm a neurologist and neuroscientist. So I see patients over at Mass General in the Memory Disorders Unit, and I specialize in early onset genetically driven Alzheimer's disease. And that dovetails with some of my research interests. So I'm a project leader for the Harvard Aging Brain Study. I've been involved in HABS since almost the beginning, not quite, but almost the beginning. And my project looks at potentially modifiable risk factors and how they alter cognitive and neurodegenerative trajectories. So we've looked at cardiovascular risk factors, how they're related to neurodegeneration, cognitive decline, and on the slide here, showing how they're related to tau pathology as well. Excuse me. And looking at physical activity, if you look on the slide, on the other side over there, we've seen that there's strong associations between levels of physical activity and the amount of neurodegeneration and cognitive decline that we've seen in HABS participants. So we've grown that into a rather large project now within HABS. I also work with the Dominantly Inherited Alzheimer's Networks on the site PI for that and on several of the steering committees and things like that for the Diane study. I also lead Sonnet, which is looking at how sleep disruption may impact cognitive decline in the buildup of tau. It's a very exciting new project. And then more recently, I've started to work on blood-based tests for Alzheimer's disease, both looking at the pathology of Alzheimer's disease and how we might measure it in blood, and then also looking at some of the mechanisms that may link cardiovascular risk factors to cognitive decline, looking at things like the VEGF pathway together with Hyun-Sik Yang, who you'll hear from in a little bit. So that's it.
Thank you. Now I'm going to pass it to Dr. Nancy Donovan.
Thanks very much. You can show me next slide. So I'm director of the Division of Geriatric Psychiatry at Brigham and Women's Hospital. I've been working in the Harvard Aging Brain Study since 2011. And I study late life neurobehavioral symptoms and their relationship to Alzheimer's pathologies and risk of cognitive decline. So in HABS, we use multimodal neuroimaging to correlate pathologic burden and other brain changes with neurobehavioral symptoms at the preclinical stage. And my work has included looking at traditional neuropsychiatric symptoms such as depression and anxiety, but also increasingly we've been interested in novel neurobehavioral changes such as declines in social engagement and self-reported loneliness. For instance, in the second image there, you can see associations of measures of loneliness in the Harvard Aging Brain Study with with tau accumulation. And looking ahead, that's an area that's really important to understand social function and socio-emotional well-being in preclinical Alzheimer's disease. So our longitudinal studies have also shown that depression and markers of psychosocial stress, such as loneliness, social isolation, and in fact, widowhood, are associated with accelerated cognitive decline. So there's a reciprocal relationship between AD progression and these neurobehavioral symptoms or markers of psychosocial stress. And indeed, these may be modifiable risk factors or targets to reduce risk of cognitive decline. And this is particularly true, we think, in people with biomarker evidence of Alzheimer's disease. So areas that are ripe for future investigation include using the HABS data set to further characterize loneliness and other subtle changes in social function in preclinical Alzheimer's disease. And then also it's going to be very important to understand mechanisms underlying women or predominantly women, especially in the Harvard Aging Brain Study sample. Because widowhood appears to be a unique risk factor for cognitive decline that's not explained by depression or cardiovascular markers, and we need to understand this better. So thanks very much for the opportunity to present.
Thank you, Dr. Donovan. So now I'm going to pass it to Dr. Jennifer Gatchel.
Thank you so much, Yakeel, and you can advance the slides. And thank you for everyone for joining the call today. I'm Jennifer Gatchel. I'm an assistant professor of psychiatry at Harvard, based at Mass General McLean Hospital. Like Nancy, I'm a geriatric psychiatrist. I'm also involved in leading neuropsychiatry efforts in the multicultural Alzheimer's prevention program at Mass General and helping to direct the geriatric psychiatry fellowship program at Mass General. So some of my earliest memories growing up were working alongside my Nana Rose and making homemade pasta and smelling the aroma of homemade gravy in our home each weekend. My grandmother was an Italian immigrant, and as I grew up, my family and I witnessed her develop the movement, cognitive, and neuropsychiatric symptoms of Parkinson's disease dementia. And although we were able to connect with a neurologist who helped to change the course of her illness, seeing her ultimately lose her battle with Parkinson's disease dementia really inspired my research and clinical research direction to pursue a career as a physician and a scientist investigating neuropsychiatric symptoms in Alzheimer's disease and related dementias. My research program, overarching research program, involves studying these symptoms throughout the dementia spectrum in terms of their neurobiology, in terms of sensitive and culturally informed assessments of these symptoms. And then finally, using this information to develop preventions and treatments that can slow cognitive and behavioral symptom progression, and ultimately leading, going back to patients and families, and how can we use this information to deliver and improve patient care for patients and caregivers. Through my work in HABS, I've been focusing on depressive symptoms in the preclinical stage of Alzheimer's disease in the relationship to cognitive changes and to AD biomarkers measured through neuroimaging. And indeed, in the HABS cohort, which is a cohort that is cognitively unimpaired and with very mild depressive symptoms, we've observed a relationship between increased depressive symptoms and increased tau in the inferior temporal lobe. And also in the second graph shown there in the middle, we found an inverse relationship between depressive symptoms and cognitive performance on a sensitive cognitive battery that detects changes in the preclinical stage of Alzheimer's disease. And that this relationship between depressive symptoms and cognition was modified by levels of brain amyloid measured by PET imaging. But finally, in more research and more recent developments, building off of these findings, I've started a research program in parallel to HABS, the Mood and Memory and Aging Study, or MOMENT study, to investigate relationships between mood, cognition, and AD biomarkers in individuals who have major depressive disorder. And also in recent collaborations, thinking about ways that we can prevent and change the course of behavioral and cognitive symptom progression through neuromodulation, and also through surveying our patients and caregivers, especially during the pandemic, to determine what are their greatest needs and challenges and how might this be impacting their future trajectory. So thank you very much, and please feel free to reach out with any questions on my email address above, and I'm also on Twitter.
Thank you, Dr. Gatchel. So now I'm going to pass it to Dr. Heidi Jacobs.
Thank you so much, Yakeel, for organizing this. Could you please show the next slide? Thank you. So my name is Heidi Jacobs. I joined the Harvard Aging Brain Study in 2015. And just like many of my colleagues, we focus on detecting the earliest brain changes that are associated with Alzheimer's disease processes. And we focus on the brainstem, in particular the locus coeruleus. And this is because, as you can see here in this row of brain slices, this is because autopsy studies have shown that one of the first regions that is affected by tau pathology early in life is the locus coeruleus, and it's Latin for blue spot. And this is why we like to call ourselves the blue spotters. Now, the locus coeruleus provides norepinephrine to the entire brain, and as such is called also a neuromodulatory subcortical system. And while we focus currently on the locus coeruleus, we are also interested in other neuromodulatory subcortical systems in the future. And we do this by focusing on method development. Examining such a tiny structure is really hard. It is small, it's deeply localized in the brain. And so we develop and we optimize methods at a 7 Tesla MRI scanner to visualize these structures. And we try to translate this to the 3T environment for clinical use. And when we can do this, we measure these properties and we relate them to other imaging markers from MRI or other PET markers, including amyloid, tau, or glucose metabolism. We also relate them to cross-sectional or longitudinal cognitive and behavioral measures. And we do this in a adult lifespan approach, given the early involvement of the locus coeruleus, with the goal to really try to understand the role of the locus coeruleus in the pathophysiological cascade of Alzheimer's disease, as well as its potential for early detection. And then finally, something that we are now moving into and we're really excited also about is that we want to use this information to guide efforts in early intervention where we are trying to delay cognitive decline. And one of these methods that we are now working with is non-invasive vagus nerve stimulation, which is a method that increases norepinephrine in the brain and also is known to enhance brainstem activity. So it's a relatively new field, and we're very excited about its potential. And if you want more information or you're interested about this, feel free to email me, or you can also find me on Twitter. Thank you so much, Yakeel, and thank you everyone for listening.
Thank you. Now I'm going to pass it to Dr. Gad Marshall.
Thank you, Yakeel. If we can move to the next slide. So I'm Gad Marshall. I'm a neurologist at Brigham and Mass General. I've been part of HABS, part of the clinical course since its inception. And like Nancy and Jennifer, I have interest in associations of neuropsychiatric symptoms in multimodal imaging in the early stages of Alzheimer's disease. I also am interested in instrumental activities of daily living and detecting the earliest changes in those in Alzheimer's disease. And recently we've had some funding to look at various imaging modalities, in particular amyloid and tau PET, as well as various MR modalities relating these particular symptoms to Alzheimer's disease. And in the past few years, we've had the opportunity to develop new assessments for IDLs. Initially, we developed a phone task that was like a phone menu or an IVRS, which individuals had to navigate in order to complete certain tasks, simulating activities that some older individuals would need to do, like refilling a prescription or calling your health insurance company to make some changes. And then more recently, we developed a smartphone app where there are a set of tasks within a patient portal, a simulated patient portal. And we abbreviated or gave an acronym to that particular assessment asset, which we're trying to validate as we speak with some additional funding. And so those have been my research interests, and I primarily have done that work at MGH. At Brigham, I run the day-to-day operations of our clinical trial unit for Alzheimer's disease, where we have a variety of clinical trials focused on either treatment of early stages of Alzheimer's, mild cognitive impairment or mild dementia, or prevention of Alzheimer's disease at the preclinical stage, primarily with biomarker positive individuals. And as part of the work at Brigham with the clinical trial unit, we also have a fellowship that we offer neurologists or psychiatrists primarily who are interested in clinical research in Alzheimer's disease, where they will get clinical training as part of our regular behavioral neurology and neuropsychiatry fellowship, as well as clinical research training in executing clinical trials, as well as investigator-initiated research, and there's crossover to the Mass General program with that. So thank you for your attention, and let me know if you have any questions.
Thank you. So now I'm going to pass it to Dr. Kate Papp.
Thanks. So you can go to the next slide. So I'm Kate Papp. I'm a clinical neuropsychologist. I work at Brigham and Women's Hospital for clinical work, and then also at Mass General for research. And I've been involved in the Harvard Aging Brain Study for a number of years. My interest really started out in understanding the interplay between semantic and episodic memory and decline in preclinical Alzheimer's disease. And then after that, I really became interested more in early detection and improving outcomes for clinical trials, both in identifying measures and ways of measuring cognition that would help in early detection, as well as tracking people more rapidly over time. So I did a lot of work with the preclinical Alzheimer's cognitive composite. And then most recently, I've been involved in digital assessment. So moving cognitive assessment from the clinic using paper and pencil measures outside the clinic to people's homes. And you can see data here on the left from in-clinic assessments in the Harvard Aging Brain Study, where we found that amyloid positives showed a practice effect over 2 years, but then they declined over time, and they're labeled in red in contrast with the amyloid negatives who are in black. So we wanted to see if we can move this annual assessment to something capturing cognitive, subtle cognitive decrements more rapidly. And so we have In the middle here, we sent iPads home and we had individuals complete the face name test each month on an iPad, and we looked at their learning curves over months. And we could again see that there was a practice effect in both groups, but that the amyloid positives in red showed a diminished practice effect. And then most recently, we've been moving this to a smartphone. So now we have individuals completing associative memory tasks and other types of memory tasks using a web-based platform on a smartphone. And we're hoping to be able to see whether or not we can look at these learning curves over days as opposed to months. And this is called the Boston Remote Assessment for Neurocognitive Health branch, which our research coordinators came up with a name. So I had no say in that. They are very creative. But we're really excited to move this digital work forward, particularly in the context of COVID, where obviously the need for remote assessment is expanded recently quite a bit. So feel free to email me if you have any questions or are interested in the work that we're doing. It's been great to be part of the Harvard Aging Brain Study team.
Thank you. I'm going to pass it to me. So I'm Yakeel Quiroz, an associate professor at the Harvard Medical School, and I have been working with the Harvard Aging Brain Study also for a couple of years now. So my research interests go in three main areas or three main directions. So the first one is that I'm very interested in studying multicultural aging. So as part of the multicultural aging, so I'm now working with the Multicultural Alzheimer's Prevention Program as the director of that program. And we have 4 studies that are going on now. So we have the Latino HABS, that is a cohort of that we are trying to recruit now participants to join HABS. We also have the Boston Latino Aging Study that is also recruiting now. We have a study with bilingual individuals to trying to understand the presentation of MCI and PPA and dementia in bilingual individuals. And recently we started with a study trying to characterize the impact of COVID-19 on the cognition, well-being and progression of AD pathology in older individuals. So this is something that we are doing with the team. So I just listed here their names. So the second main interest of mine is the biomarkers of preclinical familial Alzheimer's disease and other dementia. So I have been studying families from Colombia that had autosomal dominant AD as part of the COLBOS project, like Colombian was for Boston. And we also have studies going on with CADASIL, that is a form of vascular dementia, with autosomal dominant form of vascular dementia. And we also have familial FTD. So as part of our work with the biomarkers, so this is just to show you some of the most recent results. So we have been using tau and amyloid PET imaging to characterize the preclinical stages of autosomal dominant Alzheimer's disease. We were able to show changes in amyloid as early as they are 28 years old. They usually develop MCI at the age of 44. And we also saw changes in tau pathology in the late 30s. Most recently this year, we started publishing some of the work with plasma biomarkers, and we published some findings with NFL and with p-tau217, showing that these biomarkers can distinguish carriers from non-carriers since their early 20s, about 22, 23 years old, several decades before they actually develop MCI and dementia. And the last one that I wanted to mention here is what we have, what I call Resist Alzheimer's Project. And this started with our first case report that we published last year with somebody from these families that didn't develop MCI or dementia until her 70s, like 30 years after the age of onset. And here I just have an illustration of how much we learned from her case. Like she actually was able to inform genomics, is now informing also basic science, and hopefully in the future will inform and help us with drug discovery and clinical trials. So this is the other direction that we are going. We are now studying other escapees like her, and also like individuals that remain unimpaired until older ages for any other reason, not just necessarily for this mutation. So, and also looking for postdocs for this project and research assistants. So if you have any interest, please feel free to reach out, and also available in email and Twitter. So thank you. going to pass it now to Dr. Schultz.
Hello, I'm Aaron, and you can go on to the next slide, Yakeel. So I'm a PhD. I've been with the HABS project since the beginning as well. Currently, I'm the leader of Project 3, which is co-PI with Julie Price, who you'll hear from soon. And the focus there is on functional imaging and synaptic integrity. I'm also kind of generally known as the data person in the lab. I have a lot of interest in methods development and informatics. And I'm a bit of a pan imager, so I do a lot of work with PET, multiple modalities, including amyloid tau and FDG, amongst others, as well as a lot of MR modalities. I do have a particular fondness for functional connectivity and functional neuroimaging, and have done a lot of work with that. So I threw up a couple of quick examples of that, and also to demonstrate my fondness for a good graphic. And also do work with various types of methods development. So in the bottom right, there's an example of some work I did with Michael Perperzi, who's one of my right-hand people in the informatics group, looking at harmonization of PET tracers. And yeah, and I'm very collaborative, so I work with just about everyone in the lab. And then lastly, I wanted to embed a little Easter egg in here. So one of the things we've done most recently over the past 6 months is finalize the HABS public data release. So this is making data available, data from the first 6 years of HABS available to public data request. So if you're interested in seeing some of the data we have on offer, please visit the HABS website or get in touch. Thank you.
Thank you. So now I'm going to pass it to Dr. Patricia Benini.
Thank you, Yakeel. So I'm a cognitive neuroscientist, and I've been working in aging and Alzheimer's disease research for nearly 20 years now. I completed my PhD training at the Karolinska Institutet in Sweden before joining Dr. Reisa Sperling's lab here as a postdoc in 2008. And I'm currently an assistant professor of neurology, and I'm based both at Brigham as well as MGH. You can change the slide. So, one more, sorry. So my research has focused on the use of multimodal imaging to differentiate healthy brain aging from the earlier signs of Alzheimer's disease. And one of the research questions that have intrigued me for a long time, and I actually started working on this nearly 10 years ago, is self-awareness of memory, and especially the loss of awareness or anosognosia in Alzheimer's disease. The project has evolved to include two major questions. The first, how does self-awareness evolve over the course of Alzheimer's disease? And when can we first detect anosognosia? And I've listed some of our recent papers here on this slide. And the second research question is, which I received or was awarded an R1 for in August of last year, is to identify the neural underpinnings of memory self-awareness and the progressive dysfunction of these systems that underlies the loss of self-awareness or anosognosia with Alzheimer's disease progression. And I currently have two postdocs, Dr. Geoffroy Gagliardi and Zada Marebwa, who's working on this project. And next. The second project that I'm working on is a collaboration with Dr. Elena Rodriguez Vietes at the Karolinska Institutet, my old department, and Dr. Juan Fortea and his group at the Hospital of San Pau in Barcelona, Spain. And it's based on the recent evidence suggesting that changes in the microstructural properties of the brain's gray matter, due in part from neuroinflammatory processes, play an important role in the early pathophysiology of Alzheimer's disease. So this project uses a novel methodological neuroimaging approach using diffusion-weighted imaging, or DWI technique, to quantify microstructural changes by means of cortical mean diffusivity in the gray matter. And using this method, we are trying to understand the mechanism linking cortical microstructural changes and AD pathology, as well as its effect on brain function. And Elena presented some of these findings earlier this year at the AIC meeting. Next. And the third project is a relatively new project of mine, which aims to study the impact of stress and resilience on cognition, future cognition and brain pathology in older adults. So in April of this year, right around the peak of the COVID-19 infections here in Massachusetts, we asked participants enrolled in several of our longitudinal studies, one of them HABS, to assess their levels of stress, resilience, and stress coping strategies. And I'm currently writing up the findings from this survey. But one of the main things, and not surprisingly, we found that stress and resilience were highly related to each other, as shown in this figure here. You can reach me on this email, listen on this, if you have any questions or are interested in one of these studies. Thank you very much.
Thank you so much. Now I'm going to pass it to Dr. Yang.
Okay, thank you everyone very much for joining and actually staying throughout this call. I'm Hyun-Sik, I'm a neurologist and a geneticist, and I've been with the group for about 5 years. I first started as a hybrid clinical and research fellow. So if any of the clinical trainees in this call who are interested in hybrid training, there are many opportunities with us, so let us know. So next slide. Okay, so my research interest is in the role of immune system in early Alzheimer's disease, starting from even before the symptom onset. So genetics studies have shown repeatedly there's a central causal role of immune system in Alzheimer's disease, but it's not well understood how that actually works in humans, especially in early phases of the disease. So we want to approach that leveraging molecular data from Harvard Asian Brain Study. So starting from DNA and genes, we have whole genome genotyping data, and going on to RNA, we are actively collecting samples and are planning to generate data for a comprehensive immune gene expression profile. And then finally, about the proteins, we have recently measured a couple dozens of plasma protein markers, and we're starting to see some exciting results where new markers involved in angiogenesis and inflammation processes predicting cognitive trajectory in early Alzheimer's disease in Harvard Asian Brain Study. So we plan to integrate all these layers of data to see their association and relationship with our rich phenotype data, including amyloid and tau PET, brain structure MRI, and cognitive trajectory, to better understand what immune system is doing in early phases of Alzheimer's disease progression, and also to see whether we can find a novel biomarker that can be used in clinical research. Thank you.
Thank you so much. And that's the last one that we had for the presentation. So as I mentioned at the beginning, so HABS is a very large group, and not everybody could attend the meeting, or not everybody could present it, but this is an opportunity for you to get a better idea of what the group is doing. And now we're going to open up for some questions. So let's see if we have.
Yep, there's one, I think.
Exactly. I think that there was one for Rebecca early on. So I don't know, Rebecca, if you want to start answering that one.
Sure, I responded. I typed out the answer, but I can speak to it as well. So there was the question of what about studies that recruit participants based on their subjective report, which was a nice reminder to mention that we're recruiting for a cohort specifically for subjective cognitive decline. So an R1 in which I'm principal investigator. And so individuals, unlike HABS, where individuals didn't have to have a memory complaint to enter, we're specifically looking for people that do have a memory complaint, and then following them over time. So as a complement to HABS.
Perfect, thank you. And Jess, there is a question for you in the QA. Can you?
Yeah, so I'm looking at it now. So the question is, what data will you use within HABS to investigate the effects of sleep and cardiovascular factors? That's a good question. So there's a few different lines of data that we're bringing in to play. So with respect to cardiovascular factors, we have detailed clinical data, things like weight, BMI, medications, presence or absence of hypertension, things like that, to look at systemic vascular risk factors. So that's one approach. We're also looking at markers of vascular injury. Right now, we're looking at those in plasma and then also in novel neuroimaging. So we can look at things like white matter disruption, changes in DTI. And then as Hyun-Sik alluded to, we have a number of different markers of VEGF pathway activation and disruption of cellular adhesion molecules that we can bring to bear on this, to really ask the question of what are the mechanisms underlying the interplay of vascular factors and AD pathology in terms of prompting decline. That's the sort of question that HABS is beautifully well suited to doing. And then with respect to sleep, we're looking at sleep in a number of different ways, self-report, actigraphy, and then at-home polysomnography. So the Sonnet project that I referred to allows us to do at-home polysomnography and detailed sleep characterizations in HABS participants, and then also in a separate group of people that have subjective cognitive decline, mild cognitive impairment, or dementia. So it's multimodal and in the same way that a lot of the rest of our studies are.
Perfect, thank you. So anybody else has any questions? Any of the attendees have any questions? You can also raise your hand. I think that I'm learning how to track the hands. So you can raise your hand also. Okay, so no questions so far. So I don't know if Reisa, you want to add something? You know, we still have like about 10 minutes.
Okay. Well, first of all, it's so great to hear everyone encapsulate their research in just a minute or two, which is great. And I also want to say there's a whole lot additional research going on that we didn't even get a chance to show you because there are a few folks who couldn't be on. And again, we have research assistants and postdocs and students who are conducting a lot of research projects themselves. And so I'm so sorry we couldn't show you all of those, but I think it's a testament to the idea that, again, this really is a team, and we have people at all levels working to answer questions. And that's why we're so excited to hopefully entice some of you on here to join us. Maybe my last thing while people are asking questions is I'll just say, you know, kind of where I started. Why are we doing this? And also, one of the most exciting things for me is that what we have learned in HABS so far has actually started to first of the kind clinical trials, where we're actually bringing in individuals on the basis of them having evidence of amyloid in their brains and trying to stop cognitive decline by treating them with anti-amyloid antibodies. And now our next trial, the AHEAD study, is going down to age 55, and we'll be looking at people even before they're, quote, amyloid positive, or as I like to say, before we want to prevent them from getting a head full of amyloid. And then very excitingly, Keith and others are going to be starting a tau platform trial, again, based on this. So I just want to say that I think it's thrilling to be able to work on the fundamental science here. And what makes this group so special is also, I think, our dedication to actually using that data to really make a difference and hopefully keep people's brains healthy for a long time and eventually prevent Alzheimer's disease, because I think that's, for at least many of us, I'll speak for myself, that's the end game. And I think this work really has real life implications. So I don't know if there are any more.
Any questions?
Oh, come on, you guys on there. You must have something you want to know. You don't have to ask us science questions. You can ask us what's in the background here at all these things, and why we were talking about Rachel's hair early on.
Yeah, I know. If anybody's interested to learn more about my hair, I'm more than happy to give a platform to that.
Yeah, I second that. Just, you know, it's take it as an informal conversation, as you want to say, like an opportunity to ask questions to the group. So I know that many of you are in the junior stage, so it's an opportunity also to learn about like career opportunities or even like anything. So you have 10 minutes. So we have the first person. Yeah, this Reisa, you want to answer that one?
Let's see, under open. Okay. Yes, actually. So let's talk. So we have many CrossLab meetings, actually. So first, we have a group meeting once a week where we often have our postdocs present, and many folks here and people everyone on this call attends that, but we often have people coming in and out from other things. Keith's group also has a weekly lab meeting that unfortunately you didn't get to hear from Julie Price today, but that group really focuses on, improving imaging in particular, but that cross-fertilization is terrific. And then we have other giant meetings where we meet with not only the clinical trials group, but more recently also with people working in laboratory research with Dennis Selko's group and sometimes with Brad Hyman's group. So there's an incredible cross-fertilization. Rachel Buckley runs a journal club as well, and which has been great to revitalize. And most importantly, we have Yankee Swap once a year where everyone, all 100 of us get together. This year it's going to be a challenge to do that with COVID, but we will figure it out. I think this next one might be best handled by Jazz or Hyun-Sik about P217. Just one last thing before I bring that up. I also want to say that we are strongly committed to mentoring people. We had a fantastic mentoring retreat organized by Gad and Rebecca and many other people on this call. And so even if you do not want to join HABS, we are committed to bringing the next generation of researchers and starting from students all the way up to professors who will become interested in this field. So please do feel free to contact us.
So I can make a comment on the p-tau 217, but Yakeel, if you want to comment, please also hop in. So there's been a lot of excitement around looking at post-translational modifications of tau, especially in blood, but also in cerebrospinal fluid. So as you guys probably know, there's been huge excitement about the p-tau 181 and 217 assays in blood and CSF. So empirically, it looks like 217 might be a little bit better than 181. I think, so what I would say about this one is that I'll do a shameless plug. We have a paper in press now in Nature Communications to look at a novel fragment of tau, an N-terminal tau fragment that also has impressive predictive capabilities with respect to cognitive decline and neurodegeneration and growth of tau pathology in HABS participants. So that'll be out in a few weeks. But in general, the different phospho-tau species, it's going to be an empirical question, whether they change, which changes earlier, and are those changes uniform? So there's a whole host of different post-translational modifications that can be made to tau, and understanding how uniform those are across people and whether or not they may be sequential is going to be a fascinating story to follow, I think, for the next several years. And it's a great opportunity where we get to interface with our bench science colleagues. Dennis Selko, as Reisa mentioned, but also Brad Hyman. So we're hard at work at this. I think in the end, it's going to be an empirical question, and it may be the case that it's not going to be as simple as one being better than the other, because different people may have different balances of post-translational modifications.
Thank you, Jess. And we're going to move to the next question. So, Reisa, you want to respond?
Sure. First, hi, Toby. This is a question about collaboration with external investigators and how to actually take first steps in coordinating on collaborative research. So first of all, we definitely are interested in collaborating with other groups. And actually a lot of, I think, our most impactful papers have been across groups with Framingham Heart Study or with using ADNI or ABLE data. And I'm pretty sure Rachel Buckley may have already reached out to some of you guys for her work. So you certainly, if there's a specific thing you want to work on, you can reach anyone here, but you're more than welcome to reach out to Keith or I directly, and we can point you to the best person. Again, we've made the first 6 years of the data public. We have another 3 or 4 years that we're still collecting in people, and we're very interested in collaborating, especially with some of the really cool work you guys are doing. So please.
And one small thing there as well is on the website, and Aaron will probably speak to this, on the website, you'll probably find, is the HABS data email on the website, Aaron?
Yes, it is.
Yeah, and I think that would also be another avenue to explore. So you could email that email address. Quite a few of the faculty have access to that, and we can help to direct on or forward to Reisa for further help. So feel free to access that as well. Sorry, Aaron, if I mentioned that and you were going to say it.
No, that's great. I was going to say, definitely check out the website and see what data is available, that's probably the best and easiest way in terms of getting access to data for collaborations. There can sometimes be institutional hurdles and roadblocks to certain types of data sharing, but we're always willing to try. The other one I wanted to mention is that we're very kind of open source oriented. So if it comes to questions about methods or analytics or pipelines, we're always happy to answer questions, and we're always happy to share code.
Thank you. The next question is also for you, Risa.
Oh, about the questions about also involved in the A4 study and HABS. So the big difference, so HABS is a single site, or at least the participants come from a single site, lots of collaboration at MGH and the Brigham. A4 is in 70 sites around the world. And so there are many things you can do at one site you can't do everywhere. On the other hand, as you point out, there's a lot of similarities that we might be able to do cross data. So right now, A4 data is available actually on Loni. Aaron's been very involved in helping with that. Only the screening data, though, are available, and A4 has 4,486 PET scans for screening, so it's a magnitude larger, and we're really interested in going across A4 and HABS, and people in Keith's group have been looking at that with Tau as well. So I do think there's ways to harmonize, but there are also some real differences in terms of multi-site versus single site. And maybe the next question I can take real quick on this was about how does data get shared between the HABS investigators? With so many fantastic investigators and projects, how do you make sure there aren't too many doubled efforts? So I would say this is probably one of the things that's the hardest because it seems like to me that we should all look at data and make science together. But of course, I think we're realizing that each person needs to have a niche and something that they get known for. So in general, when two people are working on something, we try to bring them together and figure out how we can take each perspective and potentially write two papers, but with different things. But I will say this is the tension between big team science and figuring it all out, and having individual projects sometimes is tough. And on our mentoring thing, this is one of the things I want to continue to learn. But in general, I would say it's worked out well, that everybody has something they're known for. And sometimes it's the identical data, but just looking at it with a different perspective on the basis of that investigator's expertise.
And I'm also quite proud of the kind of data and informatics team for the role they play here in terms of being a clearinghouse, of being able to make lots of different data available to everyone in kind of a centralized domain. And insofar as we can, we also try to kind of facilitate collaborations and information flow between people, and it's been working really well.
And the last question is about opportunities for undergraduate research assistants. Yes, we do. We absolutely do. And I have to say, you know, if there was something that interested you on this, you should definitely, I hope, Jacqueline, how do you, if people want to actually get the slides or kind of contact someone directly, do they have that? They should, or to this website, or how could they?
I will share the link to the recording with everybody that registered, so they can have access to that information. Perfect. Thank you, everybody. Thank you so much. So we only have like 1 minute. So I just want to thank the Harvard Aging Brain Study team, the ones that presented and the ones that couldn't make it, but were with us today too. So thank you so much to the attendees. And I just want to remind you about the next two Meet the Scientist webinar in November 10, Meet the Vanderbilt team, and in December 3rd, Meet the Healthy Brain Project team. So more information in the website as well. So thank you so much, everybody, and have a good day.
Yay!